Alright, this is RNA translation. Alright, so with RNA translation, the mRNA, the mature mRNA, is in the cytoplasm, and depending on what kind of protein it's coding for, it can be either hydrophobic or hydrophilic. Now, if it's hydrophobic, it doesn't like water, so it wants to be attached to a bound ribosome, and in order to do that, it's gonna the ribosome's gonna start coding, and the code for the protein is not gonna like the water, so it'll just automatically move to a place where there is no water, and so that hydrophilic protein can continue to build. However, in this example, we're just gonna talk about free ribosome, which is ribosome that produces the protein in the cytoplasm. All right, hydrophobic. All right, so we're gonna start with a ribosome. There's two parts to a ribosome: the small subunit and the large subunit. Small subunit is what reads the mRNA 3 nucleotides at a time. A 3 nucleotide makes up one codon, so it reads one codon at a time. And then the large subunit have three three structures to it: the A site, the P site, and the E site. The A site is, you can think of it as an arrival site. This is where the tRNA that has the amino acid will be bringing the amino acid into the A site. The P site is where it bonds to the peptide bond happens, and then the E site is the exit site where the tRNA removes itself and find another enzyme and to activate it, and then the cycle starts again. So here we have a mature mRNA. It's fed through the small subunit, and as it reads, it's gonna keep reading until it comes across AUG. And once it finds AUG, the large subunit comes in, because AUG is the code for methionine. Methionine is the start codon, which knows that this is where the protein starts. This is where the important parts are. So this is where initiations start. We're gonna get the AUG codon. It matches the anticodon on the tRNA, which is UAC. So tRNA comes in with its anticodon right into the arrival site, or the aminosylrite site, comes in, and here it matches to the mRNA. So the mRNA is moved along. Here, let's take down this so it doesn't move around. So the mRNA is getting read again by the small subunit of the ribosome, and this moves along with it to the P site. Now, this leaves room on the A site for the next codon, which is ACU. ACU codes for the anticodon UGA, which is threonine. Threonine comes in, it matches with the codon, and threonine will now form a bond with methionine. So methionine detaches from the tRNA, bonds with the threonine, and moves along. Once again, moving into the next set. Now, here is where the methionine, or the tRNA UAC, reaches the exit site. The exit site is just where the tRNA is released to go wherever it needs to go. So tRNA exit, well, not yet. Well, comes in. The next triple codon or triple nucleotide is CGA. CGA codes for the anticodon GCU. GCU is arginine. Arginine comes in and matches with the codon. Now it moves, and now it forms a bond with threonine before it. So this peptide bond is formed again, that at the peptide site, or the P site. It bonds, and the the moving of the amino acid on top of each other is called translocation, right? And so now it moves again. Now the start codon can go wherever it needs to. And then the next set of codon that goes in would be CUC, which codes for the anticodon of GAG. GAG is leucine. So leucine comes in, its anticodon matches, and then it bonds with arginine, and arginine bonds with that. It makes a peptide bond or covalent bond, and then it keeps reading over, moves over. It moves over. Whoa, I lost where it was. All right. It moves over to the next one. All right. AUG, UCG, and then this one goes away. The next one comes in, replaces. Now the only reason the tRNA is coming in is because there's this free space. So the next set is GAC, which is CUG. So CUG codes for aspartic acid. Aspartic acid comes in. Aspartic acid then bonds with leucine. And it creates this bond. This comes out. This moves along this, and it moves along. And then it comes along GGA, which is CCU, CCU. Do we not have U? Oh, UCC comes in, arginine. Now arginine comes in, it moves once again, the chain translocate in, and now that you see the bond is, the peptide chain is building, it's going, and the next codon codes for ACA, which is UGU, which is 309 again, and once again, peptide bond happens, translocation, moving down onto the next one, and then the next codon codes for ACA, which codes for UGA. Next one codes for CAU, which is GUA. GUA is histidine. Peptide bond happens. Translocation. Oh dear. And as we move along near the end here, exit site, and now we have now the next codon codes for UAG. UAG doesn't code for an actual amino acid. So there's actually no tRNA that could fit into it since it's a stop codon. So the ribosome now knows to send a release factor, which then in turn releases the tRNA molecule and the polypeptide chain that we have. The polypeptide chain goes away, tRNA goes away, ribosome goes away, and the RNA is left to go find other ribosomes, and the polypeptide bond can now be folded into a 3D shape or combined